Good morning, students. In the last class we have done with your upstream processing, that is USP, upstream processing. What is upstream processing in fermentation? This is your formulating your medium, formulating fermentation medium, that is carbon, nitrogen, mineral elements, next vitamins. See here, mineral elements, vitamins. Next, what we have added? Anti-foaming agents. Next, oxygen, water. ఇంకేమేమి యాడ్ చేసాం? ప్రీకర్సర్స్, ఇండ్యూసర్స్, ఎలిసిటార్స్, next inhibitors, next cell permeable metabolites. Is it or not? All these we have added into the fermentation medium. Second, after adding all these, we have gone for sterilization by checking the pH, temperature, maintenance and sterilization. What is sterilization? 121 degrees centigrade, 15 lbs pressure for 15 minutes in an autoclave. You are going for sterilization. Now pour the sterilant. Third one, select the microorganism and load it into the fermenter. Select the microorganism and load it into the fermenter. Now this is over. Formulation of medium, formulating fermentation medium, sterilization of the medium and checking the pH and maintenance of pH and temperature and sterilization of the medium. Next, select the microorganism and load it into the fermenter. Next, this entire thing is known as upstream processing till the product is produced. Now coming to the downstream processing. That is DSP. It consists of, what are the steps that are there? First one is recovery of your product. Second one is purification of the product. Third one is checking the biological activity. Okay, checking the biological activity of the product. Next, fourth one is your recovery, purification, biological activity. Next, after checking the biological activity, we are going for packing. Next, fifth one is after packing, it is transported to consumer. అర్థమైందమ్మా? దీస్ ఆర్ ది 5 థింగ్స్. ఫస్ట్ ప్రోడక్ట్ని రికవర్ చేసుకొని, అది ఎంత వచ్చిందో రికవరీ ప్రోడక్ట్ని తెలుసుకొని, దాన్ని ప్యూరిఫికేషన్ చేసుకొని, దాని బయాలజికల్ యాక్టివిటీ చెక్ చేసుకొని, ప్యాకింగ్ చేసి బయాలజికల్ యాక్టివిటీతో కూడా లో కాస్ట్ ఉండేటట్టు చూసుకొని, తర్వాత దాన్ని ప్యాకింగ్ చేసుకొని, తర్వాత ప్యాకింగ్ అయిపోయాక దాన్ని ట్రాన్స్పోర్టెడ్ టు కన్జ్యూమర్. దీస్ ఆర్ ది థింగ్స్ దట్ ఆర్ నోన్ యాజ్ అ డౌన్ స్ట్రీమ్ ప్రాసెసింగ్. In this downstream processing, these are the steps. Now coming to the downstream processing. In this downstream processing, what we'll be doing is, see here, we'll see one by one. In this downstream processing, first is recovery. All of you know that from the outlet you will be recovering the product. Once the cells reaches, see here, for the recovery of the product, there are 3 things. One is, first is primary metabolite, secondary metabolite, third one is enzymes. Okay, these are the things that can be extracted, recovered from the fermentation unit or from the fermenter or bioreactor. What is primary metabolite? It may be amino acid, nucleotide, or protein or lipids, so or your vitamins. That means the primary metabolites are the substances, there may be carbohydrates also. Okay, amino acids, carbohydrates, proteins, lipids, next nucleotides and vitamins, these are the primary metabolites, that is nucleic acids are primary metabolites, and these are produced with the help, these are the primary metabolites, and these are produced during the exponential growth phase or log phase. When the cells are in log phase, you will get the primary metabolites. You can extract, if you are taking recovering the medium from the cultures, then you will be extracting these primary metabolites in the log phase. When you in the log phase, when you are collecting the product, you will get primary metabolites. And coming to the secondary metabolites, they are alkaloids, terpenoids, flavonoids. See here, alkaloids, terpenoids, flavonoids, all these are produced, these are the secondary metabolites, and toxins, phenols, these are the secondary metabolites that are synthesized in the fermentation medium when the fermentation medium is in stationary phase. That is, at the starting of the stationary phase, when you are collecting the medium, that is after log phase, when the cells enter into stationary phase, at that particular stationary phase, or not all the microorganisms, only that is all the microorganisms produce these things during log phase. But once the log phase, from the log phase, once the cells enter from log phase to stationary phase, all the cells are not capable of synthesizing these secondary metabolites. So only some of the microbial cells or microorganisms are capable of producing these secondary metabolites, and these secondary metabolites are produced only during stationary phase. అంటే ఏంటి, మనము లాగ్ ఫేజ్లో ఉన్న సెల్స్ని కలెక్ట్ చేసుకుంటుంటే మనకు తెలుసు ప్రైమరీ మెటబోలైట్స్ వస్తాయి అని. అంటే మనకు ప్రైమరీ మెటబోలైట్స్ కావాలి అని మనం ఫెర్మెంటేషన్ చేస్తే కనుక, మనం ప్రైమరీ మెటబోలైట్స్ కావాలని ఫెర్మెంటేషన్ చేస్తే కనుక లాగ్ ఫేజ్లో సెల్స్ని తీసుకోవాలి. అదే కనుక సెకండరీ మెటబోలైట్స్ కోసం కనుక మనం ఎక్స్పెరిమెంట్ చేస్తే, మనము సెకండరీ మెటబోలైట్స్ని ఎప్పుడు అంటే దాని మీడియం ఎప్పుడు స్టేషనరీ ఫేజ్లో ఉన్న మీడియంని తీసుకుంటాము. నౌ కమింగ్ టు ది ఎంజైమ్స్. ఎంజైమ్స్ ఆర్ ఆఫ్ టూ టైప్స్, ఇంట్రాసెల్యులర్ extracellular or extracellular. What is intracellular? The enzymes that are produced within the cell. Cell lopale produce ayyayi. Example is invertase. Uric acid. Extracellular enzymes, these are your cellulase, lipase, protease, etc. Cellulase, lipase, protease, all these are the enzymes. They are produced extracellularly, that is outside the cell. From the cell they are released into the medium. Now after all these, the most important thing is your biomass. What is biomass? Biomass is nothing but the microbial cells or microorganisms. The cells of microorganisms is called biomass, that are responsible for the production of your primary metabolites, secondary metabolites, next your enzymes, etc. are produced because of the presence of these microorganisms. And these microorganisms, after completion of your fermentation, these microorganisms are called biomass. Okay, now that is, you will be recovering the product, any of these products, based on your requirement. You will be collecting the fermentation medium along with biomass at a particular time. మీకు ప్రైమరీ మెటబోలైట్స్ కావాలంటే లాగ్ ఫేజ్లో కలెక్ట్ చేసుకుంటారు, సెకండరీ మెటబోలైట్స్ కావాలంటే స్టేషనరీ ఫేజ్లో కలెక్ట్ చేసుకుంటారు. మీకు ఎలాంటి ఎంజైమ్స్ వస్తున్నాయో తెలుసుకోవడానికి ఇంట్రాసెల్యులార్ ఎంజైమ్సా, ఎక్స్ట్రా సెల్యులార్ ఎంజైమ్సా మీరు తెలుసుకోవచ్చు. నౌ this is about your recovery. Now coming to the next one, that is purification. In this recovery only, see here, there are two methods for recovery. See here, there are two methods for recovery. They are extracellular enzymes ki, intracellular enzymes ki. That is, the product is produced in the cell, microbial cell, and these microbial cells, they are releasing the product into the medium so that you can easily collect the medium. Now what you have to do is, in this extracellular thing, you can go for the collection of your product by means of centrifugation, ultra centrifugation, ultrafiltration, etc. These are the methods by which you can go for the collection or recovery of extracellular products or extracellular substances. Now see here, before going for that, see here, this is your fermenter and you have collected the medium, fermentation medium, and this fermentation medium, spent medium, it consists of, along with medium, it consists of microorganisms. Now what you have to do is, you have to take this and into this you have to add flocculating agents. Flocculating agents are added. What are flocculating agents? Mineral salts. Any type of mineral salt is added. That is inorganic salts, ammonium sulfate, potassium sulfate, next magnesium chloride, manganese sulfate, like that. You can add any mineral salts are added, mineral salt is added, and that helps, that acts as a flocculating agent, and that will make the microorganisms. See here, when you add them immediately in the like extracted fermentation extract, to the fermentation extract, if you are adding flocculating agents, immediately the microorganisms will join with each other and form floccule like structures. See here, that means the microorganisms, the microbial cells will flock together and they join together to produce floccules. This is known as, these are known as floccules. That means the microbial cells, they join with each other. When they join together, attach or join together, then they will be forming small group of cells. Now take it and go for centrifugation. They will be collected as a pellet. This is your biomass. Biomass is collected. Again, the biomass can be reused for your fermentation, and the medium which is there, this can be used, recovered, and from that you can extract your medicine and purify the medicine or purify your product. Okay. Now this is one type. If not, simply go for filtration. When you go for filtration, take a vessel, keep a funnel, take a vessel and keep a funnel, and through this filter you allow it to pass through. That is, the membrane filters. This funnel is nothing but it is a filter sieve. Its size will be 0.5 micrometers pore size, and when the pore size is 0.5 micrometers, the microorganisms which are above 0.5 micrometers, they are collected in the sieve, and the rest of the medium that will be collected, the medium is all filtrate is collected at the bottom of the in the vessel, in the bottom vessel. Now this filtrate is taken and it is purified and the product is packed and marketed. Okay. Now see here, now this is your flocculating agents. Suppose if you are going for ultrafiltration method, instead of centrifugation and ultracentrifugation, you are going for ultrafiltration. This ultrafiltration method is useful for collecting your biomass. What is that biomass? Filamentous fungi. Okay. The biomass filamentous, if your biomass is filamentous fungi, then simply go for ultrafiltration and collect the biomass and separate your filtrate that is used for purification. This is known as your extracellular product, how they are collected, recovered. Now coming to the intracellular. See here, coming to the intracellular, this intracellular is the product that is produced within the cell. See here, intracellular means the product that is produced within the cell. అంటే ఏంటి, ఈ సెల్ లోపలే మనకి ప్రోడక్ట్ ఉంది, తయారైంది, ఇది బయటికి రావాలి, అప్పుడే మనం కలెక్ట్ చేసుకోగలము. ఫర్ దట్ వాట్ యూ కెన్ డూ ఇస్, దట్ ఈస్ ఎగ్జాంపుల్స్ ఆఫ్ ఇంట్రాసెల్యులర్ ఎంజైమ్స్ ఆర్ ఇన్వర్టేజ్ అండ్ యూరిక్ యాసిడ్. నౌ in this intracellular, for the collection of the product from the cell, cytoplasm of the cell, what are they means, there are three methods. One is mechanical, second one is lysis, third one is drying. These are the three methods. You can follow any one of the method depending upon your product, intracellular product, and you can collect the intracellular product from the cell. Now any of these processes, they will degrade or disrupt the cell wall and plasma membrane, so that the product can be released into the medium. For this, see here, in this mechanical method, take this medium and pass it through a funnel, or you can call it as a nozzle. The medium is taken consisting of your microorganisms, and this medium is sprayed through the nozzle with forcefully. That is, forcefully you are releasing the medium along with your microorganisms forcibly through the nozzle of a narrow pipe, so that it will be released like this. When it is released, due to that force, the cell gets ruptured. The cell gets ruptured because of shearing. అంటే ఏంటి, ఇట్లా చింపినట్టు ఇట్లా ఫోర్స్తో దాన్ని వదిలేసరికి దాని నుంచి వచ్చే సెల్స్ అన్నీ ఆ ఫోర్స్ తట్టుకోలేక ఇట్లా పగిలినట్టు అయ్యి, లేకపోతే పేపర్ చింపినట్టు షీరింగ్ అవుతుంది. అంటే ఏంటి, ప్లాస్మా మెంబ్రెయిన్, సెల్ వాల్ ప్లాస్మా మెంబ్రెయిన్ ఇలా చిరిగినట్టు అవుతాయి. ఇప్పుడు మైక్రో ఆర్గానిజమ్స్లో సెల్ వాల్ ప్లాస్మా మెంబ్రెయిన్ ఇట్లా చిరిగినట్టు అయిపోయి దానిలో ఉన్న ప్రోడక్ట్స్ బయటకు వస్తాయి. లైసిస్ ఈజ్, యూ కెన్ యూజ్ ఎనీ ఆఫ్ ది ఎంజైమ్స్. You can use salts, organic salts, mineral salts, or you can go for enzymes, lysis enzymes like lipases. You can use an enzyme called lysozyme, so that the cells get disrupted and release your product. The cells get disrupted and release the product. This is known as lysis, that is breaking of cells using your chemicals like your any of your salts, organic salts or inorganic salts, or enzymes like lysozyme. Now coming to the drying, take the cell and allow it to dry. When you dry, the cell wall gets distorted. That is, this water content decreases and the cell gets shrunken. Once the cell gets shrunken, still more you are drying, immediately the cell will break like this, and the contents will be released. This is known as, these are the three methods of extracellular collection, recovery of the product. Intracellular recovery of the product by means of mechanical method, or by lysis, or by drying. Okay, mechanical method, drying, or lysis, these are the three methods you can follow for the intracellular enzyme recovery. Now coming to the next one, purification. This is recovery. రికవరీ అంటే ఏమేమి చెప్పాను నేను, మనము ప్రైమరీ ప్రోడక్ట్స్ తెచ్చుకోవచ్చు, సెకండరీ ప్రోడక్ట్స్ తెచ్చుకోవచ్చు, ప్రైమరీ మెటబలైట్స్ తెచ్చుకోవచ్చు, సెకండరీ మెటబలైట్స్ తెచ్చుకోవచ్చు, ఎంజైమ్స్ తెచ్చుకోవచ్చు. ఎంజైమ్స్ అయితే ఇంట్రాసెల్యులార్ ఎంజైమ్స్, ఎక్స్ట్రాసెల్యులార్ ఎంజైమ్స్. మరి ఎక్స్ట్రాసెల్యులార్ ఎంజైమ్స్ అయితే ఎలా రికవర్ చేసుకోవచ్చు, ఇంట్రాసెల్యులార్ ఎంజైమ్స్ అయితే ఎలా రికవర్ చేసుకోవచ్చు, వీ హ్యావ్ సీన్. నౌ కమింగ్ టు ది ప్యూరిఫికేషన్. This purification is nothing but, after collecting the product, after recovering the product, you have to purify it. And this purification is not completed in this step. That is, 100% purification is not done in this purification step. But actually the purification, complete 100%, 98 to 100% purification is done in the last step. That step is known as polishing step. Now see here, in this purification step, what you will be doing is, you will be taking the product or recovered extract. This is recovered extract, and now this recovered extract is taken, and depending upon your process, that is the manufacturer is producing the product, depending upon the preference of the manufacturer, the product can be done, the product can be purified. For this purification, this consists of, the recovery extract consists of your physically and chemical related or similar compounds. See here, recovery extract consists of physical and chemical characteristics, similar physical and chemical characteristics containing contaminants. అంటే ఏంటి, వీటితో పాటు కొన్ని కంటామినెంట్స్ ఉంటాయి, అందుకే మనం ప్యూరిఫై చేయాలి, అందుకే మనం ప్యూరిఫికేషన్ స్టెప్కి వెళ్ళాలి, అందుకే మనం ప్యూరిఫికేషన్కి వెళ్ళాలి. ఎందుకు అంటే దీంతో ఆ ప్రోడక్ట్ ఎలా ఉందో ఆ ప్రోడక్ట్ లాంటి ఫిజికల్ క్యారెక్టరిస్టిక్స్, కెమికల్ క్యారెక్టరిస్టిక్స్ ఉన్న లాంటి కంటామినెంట్స్ దాంతో ఉంటాయి. అందుకోసం వీ రిక్వైర్ sophisticated, sensitive and sophisticated techniques for the sterilization of our medium. దీనికి చెయ్యాలి అంటే ఏం చేయాలి? For this you can go for your absorption. See here, adsorption chromatography, ion exchange chromatography. See here, ion exchange chromatography. Next, your ion or liquid or solid extraction, or you can go for ultrafiltration. Any of these methods, sensitive and sophisticated methods, can be selected, and you can go for the purification of your product, and this is done based on the preference of the producer. If a manufacturer is producing the product based on his interest or based on his preference, he will be purifying the product by taking any of these steps. That is, any of the method can be followed depending on the preference of the producer. The product can be purified. This is about the first step in purification. Next one, coming to the second step in purification, in this specific metabolite purification. So some of the producers or manufacturers, they are very much interested in a particular product, and that metabolite should be purified. That means a specific metabolite is collected as a product and it should be purified. For the purification of such type of specific or special type of metabolites, for purification there are two methods. One is crystallization and the second one is chromatography. What is crystallization? Crystallization is that in this method you will be adding, see here, in this crystallization method you will be taking the extract. See here, in this method you will be taking the extract. All of you know that extract means the after filtration but that is collected, the material is known as extract. Recovered material from the fermentation unit or recovered material from the fermenter is known as extract, and to this extract you will be adding potassium acetate plus alcohol. Potassium acetate plus alcohol is mixed in this extract, and that will lead to the crystallization of your low molecular weight compounds. Low molecular weight compounds are crystallized with the that is your specific compound. If it is a low molecular weight compound, that can be crystallized with the help of this potassium acetate and alcohol mixing. Next, coming to the chromatography. In this method, it is also useful for collection of low molecular weight compounds, but all if a mixture of compounds are of similar type, and a homogeneous mixture of microorganisms are there, or a homogeneous కాంపౌండ్ ఈజ్ దేర్, ఏ మిక్స్చర్ ఆఫ్ హోమోజీనస్ కాంపౌండ్స్ ఆర్ దేర్. అంటే అన్నీ ఒకలాగానే ఉన్నాయి, బోత్ ఫిజికల్ అండ్ కెమికల్గా. అట్లాంటి వాటిల్లోంచి అన్నిటికీ లో మాలిక్యులర్ వెయిట్ ఉంది, అన్నిటికీ ఫిజికల్ ఒకేలాగున్నాయి, కెమికల్ ఒకేలాగున్నాయి. అట్లాంటి కెమికల్ ప్రాపర్టీస్, ఫిజికల్ ప్రాపర్టీస్, కెమికల్ ప్రాపర్టీస్, లో మాలిక్యులర్ వెయిట్ అన్నీ ఒకలాగానే ఉన్నాయి. నౌ టు సెపరేట్ యువర్ స్పెసిఫిక్ మెటబోలైట్ ఫ్రమ్ ఏ మిక్స్చర్ ఆఫ్ similar low molecular weight, similar physical properties, similar chemical properties, from this mixture of compounds your specific metabolite can be extracted with the help of chromatography. అర్థమైందమ్మా? లో మాలిక్యులర్ వెయిట్ ఒక్కటే మన క్రైటీరియా అయితే కనుక యూ కెన్ గో ఫర్ క్రిస్టలైజేషన్. ఇప్పుడు లో మాలిక్యులర్ వెయిట్తో పాటు ఫిజికల్ ప్రాపర్టీస్, కెమికల్ ప్రాపర్టీస్ అన్నీ ఒకలాగానే ఉంటే, సిమిలర్గా ఉంటే, యూ కెన్ గో ఫర్ క్రోమటోగ్రఫీ ఫర్ ది ప్యూరిఫికేషన్ ఆఫ్ యువర్ ప్రోడక్ట్. నౌ కమింగ్ టు ది నెక్స్ట్ వన్ ఈజ్ యువర్ డీ వాటరింగ్. ఇక్కడ రాయలేదు నేను. వాట్ ఈజ్ డీ వాటరింగ్? ది ఎక్స్ట్రాక్ట్ ఆర్ యువర్ ప్రోడక్ట్ or your microorganism, whatever it may be, they consist of water, because till now they are in the fermentation unit consisting of water. As I told you, in upstream processing we will be adding water into your culture medium. Now see here, due to the presence of this water in them, their volume will be increased. They have increased volume, అంటే ఎక్కువ వాల్యూమ్ ఉంటుంది వాటికి, అంటే సైజ్ ఎక్కువగా ఉంటుంది, లేకపోతే ప్యాకింగ్ చేయడానికి లేకుండా చాలా ఎన్నో క్యాన్స్లో పోసేటట్టు అంత ఉంటుంది. సో వాట్ యూ హ్యావ్ టు డూ ఈజ్ యూ హ్యావ్ టు గో ఫర్ డీ వాటరింగ్. దిస్ డీ వాటరింగ్ ఈజ్ డన్ బై డ్రై vacuuming or vacuum drying. First one. Second one is spray drying. Third one is your lyophilization, that is freeze drying. There are three methods for dewatering. Vacuum drying, that means you are using high heat and heat and vacuum are created. That is called as vacuum drying. Spray drying means you will be heating the compound at 150 degrees to 250 degrees centigrade. Hot air is passed through your product so that the product gets dried. Coming to the next one, freeze drying. Andarki telusu. Ee freeze drying means I have explained in your previous classes, that is lyophilization. In this, along with that is a solid material is converted, sublimed to gas. So the solid material is converted to gaseous mixture. So this is known as your sublimation, and the sublimation process is taking place in the freeze drying, that is nothing but your lyophilization. In lyophilization, this is the method you can go for your freeze drying. This is the dewatering is done. అంటే దానిలో ఉన్న ప్రోడక్ట్లో ఉన్న నీళ్లు తీసేయడానికి, నీళ్లు ఉండడం వల్ల అది ఎన్నో క్యాన్స్ నిండా పోయాల్సి వస్తుంది, దాన్ని స్టోర్ చేయడం కష్టము. అందుకోసమని ట్రాన్స్పోర్ట్ చేయడము, దాన్ని హ్యాండిల్ చేయడము, ట్రాన్స్పోర్ట్ చేయడము, స్టోర్ చేయడం అన్నీ కష్టం కాబట్టి దానికి డీవాటరింగ్ చేయాలి. ఆ డీవాటరింగ్ ఎందుకు చేయాలంటే దాని వాల్యూమ్ ఎక్కువ ఉంది కాబట్టి తక్కువ చేయాలి. తక్కువ చేయడానికి ఎనీ వన్ మెథడ్ కెన్ బి ఫాలోడ్. Of all these methods, this method is very beneficial, that helps in spray drying, helps in rapid drying of our product. Now coming to the dewatering అయిపోయింది. Next last step is polishing. What is polishing? Extra purification of your compound, that is 98 to 100% purity. That is 98 to 100% purity of the product can be maintained with the help of polishing. That is extra purity is maintained, and this helps in the marketing and transportation and marketing of our product. What you will be doing is you will be purifying it. Extra care will be taken for purification, and again you are going for ion exchange chromatography or your adsorption chromatography, and you are purifying your compound, so that again you are going for chromatography. This helps in 98 to 100% pure compound production. And this pure is packed, and it is transported, and it is stored. That means the product after polishing, that is again going for chromatography, 98 to 100% compound is purified, and that purified compound is packed, transported and stored, or packed, stored and transported, whatever it may be. It is reaching the consumer and its cost will be low cost. ఓకే, ఇప్పుడు ఈ పాలిషింగ్ చేసినప్పుడు ఒక్కొక్కసారి ఏం చేస్తాం, పాలిషింగ్ కాదు, ఇక్కడికి వచ్చాను నేను మళ్ళీ. ఈ ప్యూరిఫికేషన్ చేసినప్పుడు, ఈ ఫిజికల్ కెమికల్ చేసినప్పుడు దీనిలో ఏం చేస్తున్నా అంటే ఐ విల్ బి యాడింగ్ ఎక్సెపియంట్స్. ఇక్కడ యాడ్ చేస్తాము పాలిషింగ్ దగ్గర. ఐ విల్ బి యాడింగ్ excipients. They are nothing but excipients are nothing but your cheap, you can call it as cheap compounds which are not reacting with our product. Such type of cheap material is added, that material or cheap material is added. This material will help in the purity of our compound, and they also help in the decreasing the cost of your product. Not only that, they are useful for packing, and after packing they can be stored along with your product and they are transported. So this is about your downstream processing. So with this we have completed our second unit, that is fermentation medium, types of fermentations, that is submerged fermentation, next is solid state fermentation and submerged fermentation. దానిలో మీకు ఏమని చెప్పాను, బ్యాచ్ కల్చర్, కంటిన్యూస్ కల్చర్ చెప్పాను. బ్యాచ్ కల్చర్, ఫెడ్ బ్యాచ్ కల్చర్, కంటిన్యూస్ కల్చర్, ఏరోబిక్ ఫెర్మెంటేషన్, ఎనరోబిక్ ఫెర్మెంటేషన్ వీ హ్యావ్ కంప్లీటెడ్. నౌ ఫార్ములేషన్ ఆఫ్ మీడియంలో ఏమేమి యూజ్ చేసామో చెప్పాను. కార్బన్ నైట్రోజన్ సోర్సెస్ స్పెషల్గా చెప్పాను. తర్వాత ఆ రెండు మీకు లాంగ్ క్వశ్చన్ కానీ షార్ట్ క్వశ్చన్స్ కానీ వస్తాయి, కార్బన్ నైట్రోజన్ సోర్సెస్. ఇది డౌన్ స్ట్రీమ్ ప్రాసెసింగ్ ఎట్లా రికవర్ చేసుకుంటాం. ఈజ్ ఇట్ ఓకే ఆల్ ఆఫ్ యూ? సో టుడే ఐ విల్ స్టాప్ హియర్, and in the next class we will meet with the third unit. Thank you students.